Okay. There we go. All right. So this is week 2. We're talking about enzymes and metabolic reactions. So related to your metabolism. So we have different kinds of enzyme reaction, metabolic reactions. We have anabolic reactions and we have catabolic reactions. Anabolic reactions build or unify molecules. So they build or they put molecules together. And we have a little picture of that. We have a molecule. We have some ATP. We have another molecule. And they build a bigger molecule. In a catabolic reactions break down molecules. So we go from the product AB to the individual reactants. Okay, scroll up so I can see. Both require an initial input of energy called the energy of activation. There are 2 natural ways to get this initial input of energy. One is raising the temperature, and another one is through physical movement. And this graphic on the side shows you that if you have a substrate, which remember are the components of a chemical reaction, that if you have a substrate and you don't have an enzyme present, it takes a lot more energy to eventually activate that. You need a lot, you work a lot harder. The cell works a lot harder, or the molecule works a lot harder without an enzyme present to accomplish the same reaction. With an enzyme, you don't need as much energy to start a reaction. An activation energy with an enzyme is in purple, so you can see how much lower the peak is, versus without an enzyme in the blue. Now, what is an enzyme exactly? Well, enzymes are a class of proteins. They're globular proteins that serve as catalysts. So catalysts, remember, are substances that influence the speed of reactions. They can slow them down or they can speed them up. Usually they speed them up. So they increase the rate of a reaction and they function repeatedly. So you have a substrate. That's what our reaction is made from. Those are like our cooking ingredients that attaches to an active site on an enzyme. And then you have this enzyme substrate complex, because now you have this enzyme with the substrates attached to it that then gives you, that leads to a product, because remember the substrates come together to make the product. And then after that happens, the enzyme detaches itself. So that's how that comes about. They do not alter the nature of the reaction, and they decrease the energy of activation needed for a reaction to occur, which is just what I was showing you earlier about the blue wave versus the purple one. When you have an enzyme, your activation energy is a lot lower. Enzymes act as organic catalysts. Remember, organic substances are those that contain carbon, hydrogen, and oxygen. So a catalyst that is a substance that speeds up the rate of chemical reactions. They control how quickly the reactions occur. They maintain our ability to survive and to function. And there's two kinds of reactions. So there's organic and inorganic catalysts, okay? Organic catalysts are comprised of organic compounds often containing carbon, okay? Now, their mechanism of action involves covalent bonding. So covalent bonding, remember, means that the atoms share electrons, covalent. So they have those shared electrons usually in the last valence shell. The stability is that the reactions are, or the enzymes are more sensitive to what's going on based on the temperature or the pH, the acidity level. Inorganic catalysts are made from inorganic materials like metals or metal oxides. Those mechanisms of action are more stable at high temperatures or under extreme conditions. The chemical reaction in our experiments. So apparently you did a lab and you have a general enzyme catalyzed reaction. So you have substrate molecules that in the presence of a temporary enzyme substrate complex produce an enzyme molecule. Now, what are you creating today? Well, you're creating 2H2O2 and it reacts to become 2H2O plus O2. So turnip juice has a specific peroxidase. So H2O2 is peroxide. You have two molecules of peroxide that decompose into two molecules of water, to H2O, this is water, two molecules of that, and one molecule of oxygen. So again, this picture on the right is just a picture of what the words tell you on the top. You have a substrate, which remembers the enzyme, and then you attach the substrate to it. Then they bind to an active site, and then after the reaction, the products are produced and the enzyme detaches. Okay, so this is a picture of the same thing. And so here, what is the substrate? And what is the enzyme that you have? What should we be looking for in today's experiment if this reaction were done in a flask test tube? Products form what? Okay, let's see. So enzyme characteristics number 1, the morphology. And in this case, it's talking about the function of an enzyme, and it's dictated by its structure or shape. So morphology refers to the structures. If we were talking about morphology of language, we would be talking about the vocal structures that help us to create verbal language. Morphology here is the structure or the shape of the enzyme. And enzymes are globular proteins. Each enzyme has a characteristic 3D shape with pockets that serve as active sites. The reactants are called substrates, and they fit into the active site like a key into a lock. So our catalase, which is our enzyme, our catalase is specific to breaking down H2O2, which is peroxide, okay, but nothing else. Mechanisms of enzyme activity number 2. So the first one was morphology. This one is temporary bonds. When a substrate binds to the active site of an enzyme, if it is compatible, it will form temporary bonds with the enzyme. Part of the enzyme temporarily binds part of the substrate molecules. What does this mean? Are enzymes used up in the reactions they catalyze? Usually enzymes are not used up. You can reuse the enzyme over and over because all they're doing is influencing the speed and the activation energy that you need for your reaction. So you have substrate A, substrate B, that go into active sites on the enzyme substrate complex, and they produce products. Naming enzymes. Here's a little, it's like a little aside about naming it. The first enzymes discovered were given arbitrary names. That means they didn't have a pattern. People just named them however they wanted. But an international committee later decided to end all enzymes with the suffix ase, ACE. They also decided to make the first part of the name apply to the function of the enzyme. So phosphate aces, phosphatases. Phosphate is this first part of this word. Phosphate removes phosphate groups. Phosphatase removes phosphate groups. Synthetases catalyze dehydration synthesis. So here we have a synthesis reaction. It's dehydration synthesis. So synthetases speed up dehydration synthesis. What does DNA polymerase do? Well, that would mean that it would speed up reactions of DNA polymers. What does RNA polymerase do? It would influence or speed up RNA polymer reaction. So that's how that works. Those are some examples. So peroxidase, which is today's reaction, would break down peroxide. Enzymes act by reducing the activation energy, which increases the rate of reaction. An uncatalyzed reaction, so this is another picture of the same picture that we saw a few slides back, except instead of being the purple and the blue, this is now blue and red. So if you don't have an enzyme present, you need a higher activation energy. This is where the blue curve EA comes in at the top. If you have a catalyzed reaction, then it's going to decrease the activation energy that you need. So an uncatalyzed reaction requires a higher activation energy than does a catalyzed reaction. And then go ahead and scroll up so I can read you the bottom part. There is no difference in free energy between catalyzed and uncatalyzed reactions. So once you have the reaction, whether it has an enzyme or it doesn't, the free energy that's released is the same. Okay, so here's some examples. Ether, chloroform, and other drugs. These are, we still use some of these in different forms, but ether and chloroform are used as general anesthetics. So ether, chloroform, and other drugs inhibit enzyme activity by binding to the active site. So these elements bind to the active site so that the enzyme can't bind on the site that can't act in the reaction because it can't bind to the active site. So it's not able to bring the substrates together. They inhibit enzyme activity by doing that. Okay, so once we do have an enzyme reaction, how do we measure the activity? Well, it's measured by the rate at which the substrate, remember that's the initial ingredients, are converted to product. And that's influenced by some things that we've already seen: temperature, pH or acidity. Acidity, if it's less than 7, baseness, if it's larger than 7, the pH. The concentration of enzyme and substrate, how much enzyme you have compared to how much substrate ingredients there are, and the concentration of cofactors and coenzymes. There's possible simulatory or inhibitory effects of products on enzyme function. So it's possible sometimes for products to stimulate or inhibit the effects of products. So sometimes the products that you produce change how the enzyme activity is. So we're going to look at effects of temperature first. An increase in temperature will increase the rate of reaction until the temperature reaches a few degrees above body temperature. At this point, the enzyme will be denatured. So typically, a slight increase in temperature will help to speed up reactions. But if it gets too high, and here I believe he's talking about reactions in the body, because remember, we're talking about organic enzyme reactions. So if you get too hot, then it's going to denature, because remember that organic enzyme reactions don't do well at extremely high temperatures. So we had that before. High temperature will typically denature enzymes and proteins in general. This changes their shape and destroys their function. So enzymes may not get used up in the reactions, but they can denature if the temperature is too high and fail to work. Okay, so what about pH? Well, enzymes inhibit peak activity within a narrow pH range called the pH optimum. Due to changes in enzyme conformation, the extremes in pH can denature enzymes. Not all enzymes like pH 7, not all enzymes like neutral conditions. Some enzymes like acidic conditions, some enzymes like basic conditions. Optimum pH reflects the pH of the fluid or the environment that the enzyme is found in. And there's a picture of that on the right-hand side. If you scroll up a little bit, it'll show you pepsin is an enzyme that's in your digestive tract where your acidity is typically higher. So notice it typically becomes very effective around pH 2 or 3 and drops off as you salivary amylase that's in your mouth. Your mouth is slightly acidic, but it's more neutral, has the highest effectiveness when you're at a pH of 7. But something like trypsin prefers things that are basic. So it relates around like a 9. Why do you think each of these pH enzymes have a different pH optimum? Part of it is related to where it's located. Where it's located in your body, and what it's designed to do. And part of it is related probably to its structure, to its format. Okay, so what about substrate concentration? Well, if you get a very concentrated substrate to start with, the enzyme becomes more saturated with substrate, which makes sense because if you have a whole lot of ingredients to start with, your enzyme that's going to increase this reaction is going to become saturated with that. So that happens. And then once all the substrate is bound, the rate of the reaction plateaus. So the more substrate that you have to bind to a receptor, the more ingredients you have to process, you can speed up that process, yes, but you still have more substrate. Right? So it's going to keep processing substrate until it's all bound to the receptor site. And then the speed's going to plateau. Okay, now what are coenzymes? Okay, so coenzymes are additional small molecules. They're like enzyme helpers that help the enzyme do its job. Coenzymes are organic molecules. So organic molecules made from water-soluble vitamins. Okay, not all vitamins are water-soluble, but they're made from water-soluble ones. Okay. Now you have your lab set up here. You put the water in there, you do the whole, put the function. Then you do okay, so you did the pH, you did the temperature, and then he did the substrate part. Right. So that was your experiment there. Okay. Okay. So what are we gonna work on next?
And then for my speech class,
Okay.
so I have to find two like job postings. So I was able to find the first one, which was like
It
position.
says you'll create it. Doesn't say you'll find it. You'll create it. This says you will create it. So you're writing it, aren't you?
Yeah,
Okay. And you're looking for a professional. Okay. So you need to create a posting for a position at a private practice. Well, let's model it after something that we already did. Let's see if I can find an example of SLPA position, job posting, job posting in private practice, California. Just so we can see some examples. Uh, okay, let's take a look at a couple of these and see what they look like. Um, so here's one. Let me share the screen. Let me go over here. Then we can start to um, get some ideas. So this one says, salaried clinic-based SLPA. Previous experience in speech pathology or other related field. Familiarity with communication disorders. Ability to build rapport with patients. 401k, okay. Speech language pathologist assistant. SLPA will provide speech and language services for pediatric to school-age population under the supervision of a speech language pathologist. With a focus on individuals with developmental disabilities, our therapists utilize the latest in evidence-based practice to provide stellar assessment and therapeutic services. Flexible schedules for both full and part-time staff. And so here's your benefits, here's your responsibilities. develop, follow therapy and treatment plans for speech disorders, implement standardized tests for accurate diagnosis of communication disorders, establish treatment goals and milestones with patients, document and store treatment plans and progress reports, and work collaboratively with other professionals. So this, I guess these people work for like they probably go in and contract with school because it's a 10-month position. Let me see if there's um something else that gives us some. Yeah, there's not a lot that's available. Um, but let's see if there's something else that I can. Yeah, there aren't too many right now. Um, can look at the forums and see if there's no, this person's talking about let me see if I can find use different search terms. There's not a lot out there. Okay, I'm not seeing too much as far as, but what is it that she wants in the actual, it says create the post, but what does she actually want in it?
She just like wants us to like include mm, like what setting the position is located, client population, SLPA will be working with three main responsibilities for the position, educational qualifications and licensing qualifications.
Okay, so something like, okay, put up your other one that you did with the Google Doc so I can follow the, because it's pretty much the same. It's just gonna do your
Yeah.
setting is just gonna change. Um, so um, okay, let me see if I can do it like this. I'll put it in the chat so I can like um so you can say, I don't know, like um you can make up the name of it. Like you could say um check the chat. Um, that's more than A, I think. Um, 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 oh, we have to say what they do. Okay, so, Mm. Give me a minute here. I'm gonna look something up. Um, okay. Am I missing anything?
Mm, I don't think so.
What is the position and client position? Three main responsibilities. The main responsibilities go in the middle. They're under C. Um, so the main responsibilities, yeah, so the main responsibilities would be under that. And then according to your pattern, and then the qualifications and the licensing, um, would go last. That still goes, but that's not C. That's a different. I think it's just a matter of the order of some of it. I think it's there, but I think the order is a little bit off based on what I saw on the. And then it tells you what we treat, so that comes, the treatment population comes sooner, doesn't it? Because I have that at now it's at the bottom. You see it?
Yes.
So that's actually the second, that's before you get to the.
I think that's it for today.
Okay, Daniela. Then I'll see you later this week, okay?
Okay.
All right, take care.
Okay, so thank you.
You're welcome. Have a good weekend. Bye.